Historically, the science of vaccinology has relied on a straightforward empirical method. Take a pathogen, kill it or weaken it, and introduce it to the human body to generate protective immunity. This table details the specific organisms we have successfully built vaccines for. It is a dense record of development, extending across centuries of medical research. But look closely at the summary data here. Of the more than 400 known human pathogens, empirical methods have only yielded vaccines for 36 of them. There is a massive gap in our defensive capabilities. To close that gap, researchers proposed rational vaccine design using immunoinformatics. The theory is precise. Sequence a pathogen's genome, compute the exact antigenic targets on a computer, and synthesize a peptide sequence that acts as a custom-built vaccine. The math is elegant. The clinical reality in the human population is entirely different. Over 100 synthetic peptide vaccines have reached Phase 1 clinical trials. Roughly 30 progressed to Phase 2. Exactly 0 have passed Phase 3 to become available for human use. We can sequence a pathogen's entire genome flawlessly. We still lack the capability to compute how that sequence will actually behave inside the biological environment of a human host. This slide defines the specific computational target required to make a synthetic vaccine work: the epitope, the quantum unit of immunity. An epitope is the exact physical surface on an antigen where an antibody or T-cell receptor binds. Linear epitopes consist of short, continuous amino acid sequences, easily represented as one-dimensional text data. But conformational epitopes depend entirely on the folded, tertiary structure. Amino acids sitting far apart on the initial sequence fold together to form a single binding surface. The computational challenge is selecting a specific 9 to 15 amino acid sequence out of a massive pathogen genome, confident that it will fold correctly and trigger an immune response once synthesized. Identifying foreign DNA is a straightforward data parsing task. Identifying a functional epitope is a complex geometric and biological prediction problem. This diagram outlines the immune pathways. To understand the prediction bottleneck, we have to look past the humoral, or antibody pathway, and focus entirely on the cell-mediated response, shown here on the right. For an MHC Class 1 response to occur, a computer model cannot simply look for a sequence match. It has to simulate a rigorous 3-stage biological filtration process. Stage 1 is proteasomal cleavage. The algorithm must accurately predict where the cell's own internal machinery, the proteasome, will chop up a foreign cytosolic protein into smaller fragments. Stage 2 is transport. The model must calculate the binding affinity of those specific fragments to TAP transporters, which act as membrane gates, allowing the sequence into the endoplasmic reticulum. Stage 3 is MHC Class 1 binding. The model must determine if the remaining fragments possess the exact kinetic requirements to bind securely to the MHC molecule and remain attached for days. If a sequence fails at any one of these 3 independent steps, the entire prediction yields a false positive. This strict biological pipeline explains why initial bioinformatics models generate so many errors. Early immunoinformatics researchers attempted to conquer this filtration pipeline using sequence-based machine learning approaches, like Gibbs sapling. They trained the algorithms by feeding models massive matrices of known 9 amino acid peptides, each paired with their empirical numerical binding scores. Tools like hidden Markov models and support vector machines analyze these sequences to identify patterns and draw statistical decision boundaries. This chart shows the metric used to score these models: the receiver operating characteristic curve, which plots the true positive rate against the false positive rate. The area under the curve, or AUC, defines the model's accuracy. When evaluated, the best sequence-based models achieved AUC values of 0.83, while others dropped all the way down to a perfectly flat 0.5. In clinical statistics, an AUC of 0.5 means the algorithm is performing no better than random chance. Sequence-based models have hit a mathematical ceiling. A one-dimensional text array cannot capture the biological context needed to guarantee a reliable prediction. The data also has to account for 2 massive biological complications. First, pathogens like HIV mutate constantly, specifically to escape proteasomal cleavage and TAP binding. Second, human MHC genes are hyperpolymorphic. There are over 2,290 known human MHC alleles. Predictively, the cellular receptor is a moving target across the population. This is compounded by heterozygosity. Every normal human cell expresses 6 different MHC class I molecules, derived from 3 separate parental genes. Additionally, every individual human has a unique T-cell receptor repertoire containing roughly 10 million variations, heavily dependent on their personal disease and vaccination history. Designing a universal synthetic vaccine using pure sequence data is currently impossible. An epitope sequence that binds perfectly to one MHC allele in the lab will fail completely across the rest of the genetic population. The problem scales further when we shift from CD8 cells to the CD4 T helper response, which relies on the more intricate MHC class II pathway. Unlike the class II pathway, the exact biological processing for class II proteins inside the cellular endosome remains an open mystery in biology, preventing accurate upstream modeling entirely. This model illustrates the critical geometric difference. The MHC class II binding groove is physically open at both ends. Because the groove is open, the peptide rests inside with its ends hanging out. These overhanging residues sit outside the core sequence, but they physically alter the conformation and affinity of the internal bond. A one-dimensional sequence model that only analyzes the core amino acids has no way to calculate the physical drag or the structural shifts caused by those flanking overhanging pieces. A 2007 study by Wang and colleagues laid out the exact cost of this missing dimension by evaluating sequence-based predictions across 16 different MHC class II alleles. The accuracy was poor. Pure sequence methods successfully recognized the binding core between 21 and 62% of the time. One specific method, called tapetotope, stood out from the rest. In the same evaluations, it achieved 100% core recognition. Tapetotope succeeded because it integrates exact three-dimensional structural information from X-ray crystallography rather than relying on sequence motifs alone. To break through the current accuracy ceilings, predictive algorithms must map the physical geometry of these molecules. The future of synthetic vaccines requires us to move past sequence matching. We have to embrace the computational weight of modeling a fully three-dimensional, in silico immune system.